Hello, hi. Hi students, I think I'm audible. Hi students, do let me know in the chat section, am I audible and visible to you all? Yes, am I audible and visible to you all? Hello? Students, am I audible, visible? Okay, I think I am. So hi everyone, welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma. I hope I'm perfectly audible and visible. Yes, I hope I'm perfectly audible and visible. Okay, so guys, welcome to the class. Today we are going to complete biological classification. It's the part 2 of this chapter, and today we will complete monera and protista. And if you people will support, definitely we'll finish fungi as well, right? But here it's not our target that we have to complete the syllabus early. Our target is to deliver the appropriate content, to discuss the NCERT, to clear your concepts, right bache? And for that, right, even if I have to take another part, we are okay with that, right? We are okay with that. Yes. So tell me, how's your preparation going on? Revising properly or not? Revising properly or not? I'm so sorry students, I was not able to take class on Tuesday and Wednesday due to personal reasons. So that's why I'm taking the class today. And tomorrow also there will be the class if we'll finish the biological classification today. Okay, so then from tomorrow we'll start the plant kingdom. It's fine now. Let's finish the diversity first. Yes? Tell me. Let's finish the diversity first. What says students? Okay, so let's continue, right? Let's start the kingdom monera. And in the last class, if I have to summarize, in the last class, right, we started the monera, but we discussed the structure of bacteria, isn't it? We discussed the structure of bacteria. So I hope the structure of bacteria is clear to you people. And I gave you the homework that what is gram-positive bacteria and what is gram-negative bacteria. So anyone in the class who can answer this question? Gram-positive and negative bacteria. Anyone in the class who can answer this question? Gram-positive and negative bacteria. Yes? Gram-positive and negative bacteria. So that's what you have to answer. Okay, that was your homework. Okay, so now let's continue students, and before that let me tell you, because you know that Independence Day is coming, so there is a special offer for all of you, right? Yes, there is a very special offer for all of you, and here you guys can see it is the early freedom sale offer, right? It is the early freedom sale offer, so you can purchase any NEET UG subscription, and yes, you will get a chance to get 100% cashback, and that is for 10 lucky winners only. So I'll say, because this offer is starting from today itself, so there is a link in the description box. Click on that link and please join this, right? Join this, right? If you will be among that top 10 lucky winners, you will get 100% cashback. Imagine, you will get 100% cashback. Okay? You'll get 100% cashback. And second thing is free flashcard for your quick revision for 20 lucky winners. Okay students, and this offer is valid from 8th August to 10th August. So it has just started. It has just started. So join the batch now. So there are 2 batches for all of you, right? One is for 2026 aspirants, right? One batch is for NEET 2026 aspirant. If you are targeting NEET 2026 and if you want to learn from us, that is from Team Avengers, then bache, there is a Nimbus Pro batch for all of you. Nimbus Pro Reloaded batch for all of you. Yes, it is a live batch. Okay, and for that again, because there is a price drop, so you will get it at rupees 4,999. Okay, you will get it at rupees 4,999. And secondly, obviously your Phoenix Pro batch, which has just started, which is the last fully live batch for the NEET 2025 aspirants, and it is again at rupees 4,999. And you will get the access of all India test series if you enroll in this particular batch. So as I said, you can get the 100% cashback plus a chance to win the flashcards. So without wasting your time, because the offer has just started, okay, offer has just started, so you can enroll in this. Okay, enroll in this. Done? Okay, so let's start the class. Let's start the kingdom monera. So start with the quick revision. So in the last class we have discussed the biological classification. We know that who attempted to classify the organism first. You know that Aristotle is the name. Okay, scientifically he tried to classify the organisms first, right? And he divided the organisms into two categories. One is your plants and animals. So plantae and animalia, or you can simply say plants and animals. Okay, plants and animals. So he tried to classify the organisms, but actually that proper kingdom system, right, it was firstly used by Carolus Linnaeus in the 2 kingdom classification system. Then 3 kingdom is also there, 4 kingdom is also there, 5 kingdom classification is also there, and 6 kingdom classification is also there, right? So here you can see the first attempt made by Aristotle, 2 kingdom classification Carolus Linnaeus, 3 kingdom classification Ernst Haeckel, 4 kingdoms classification Copland, 5 kingdom classification R.H. Whittaker, and 6 kingdom classification it was given by Carl Voss. So that is what we have completed in the last class. Is that clear, students? Is that clear? So this is what you need to revise quickly. So can you tell me, in the 6th kingdom classification system, what was new? What was new? Yes, bache. Tell me quickly. In the 6th kingdom classification system, what was new? Tell me. Yes, tell me quickly. What was new in the 6th kingdom classification system? Quick. Yes. What he introduced? Carl Voss. Anyone? 6 kingdoms were there, but exactly. So domains. Right, domains, they were introduced here. So 3 domains were there. Can you name that 3 domains? Yes, students, can you name that 3 domains? So 3 domains were there. So that 3 domains, they are archae, then bacteria, then it is eukarya. Archae, bacteria, and then it is eukarya. These are the 3 domains. So basically Carl Voss, he divided the kingdom. He divided the kingdom monera into 2 different kingdoms. One kingdom contains the Archaebacteria and another kingdom contains the eubacteria. So that is what we should know. Okay, that is something we should know. So now let's start this kingdom monera. So in the last class, students, we have already discussed. Yes, so when we talk about the kingdom monera, you know that the sole member is going to be bacteria, isn't it? When we talk about kingdom monera, we know one thing, that we are going to talk about. Yes, one second. Okay, so we are going to talk about bacteria. Am I right? We are going to talk about bacteria. I hope you guys are aware of it. Yes, I hope you people are aware of it. So basically bacteria will be there. What will be there? Bacteria will be there. So we know that it is going to be a very basic structure. It is a very basic cell. Right, the genetic material will directly be there in the cytoplasm, and we are going to call it nucleoid. Am I right? Plus, the membrane will form some extensions that are known as mesosomes. Right, that are known as mesosomes. When we talk about the organelles here, you will only see a non-membrane bound organelle, that is ribosome. Right, that is ribosome, and that is also 70S ribosome. What is this S? It is the Swedberg unit. Right, it is the Swedberg unit. It is a sedimentation coefficient. That is also PYQ. Okay, that is also PYQ. So it's the ribosome, 70S ribosome, having 50S as the larger subunit, 30S as the smaller subunit. So there are some storage granules also, like inclusion bodies. Remember, inclusion bodies. These are the storage granules. They are going to store the food. Okay, and other than that, we'll see that in a bacterial cell, to one mRNA, many bacteria, they are attached, and we will call it as polysome or polyribosome. Okay, we will call it as polysome or polyribosome. So that is also we have discussed in the last class. So if I talk about the coverings, cell membrane, then there is cell wall. So can you tell me about the cell wall? The cell wall is made up of, yes students, the cell wall is made up of, anyone in the class? I'm just revising this. So there will be the cell wall, and we know that that cell wall is made up of peptidoglycans. Okay, that cell wall is made up of peptidoglycans. We call them as murines. Are you getting my point? We call it as murines. So peptidoglycans are there, and we call it as murines. What is it? It is murines. Understood? Understood? And there are some short peptides also. Then there is a glycocalyx, which is the cell coat, which can be solid and which can be loose sheath-like. So that's something we have covered in the last class. And moreover, in the case of bacterial cell, you will see plasmid. That is extra chromosomal DNA. Okay, that is extra chromosomal circular DNA that can replicate by its own. So this is a basic structure of a bacterial cell, a prokaryotic cell, and it belongs to kingdom monera. So overall in kingdom monera, what we need to focus? Obviously, we need to know about the types of bacteria. We will discuss Archaebacteria and the eubacteria, right? We are going to discuss Archaebacteria and eubacteria, right bache? Archaebacteria, the primitive one, eubacteria, true bacteria. We will also discuss about the nutrition in the bacteria. We'll also discuss their habitat, right? We will also discuss the way by which they will reproduce. So these are the things that we are going to discuss here. Okay, so now the homework that I have given you, right? So obviously in the chapter cell we discuss it, but still let's relate this topic here. Gram positive bacteria and gram negative bacteria. Gram positive bacteria and gram negative bacteria. So students, we categorize bacteria like this on the basis of their cell wall, right? Actually, there is a stain, right? There is a stain by which we can color bacteria. That is the gram stain. That stain is given by Kristen Gram. So if any bacteria, it shows the color with gram stain, that will be gram positive bacteria. If it doesn't show the color with the gram stain, it will come under the gram negative bacteria. Again, I'm repeating, if it will show the color with the gram stain, it comes under gram positive bacteria. If it is not showing any color because of this gram stain, it will come under gram negative bacteria. So why is it so? On the basis, the difference is on the basis of the cell wall. So in the case of gram positive bacteria, the cell wall is very thick. Okay, it's thick. It's very thick, having peptidoglycan, having teichoic acid. But here in the case of gram negative bacteria, you will see that cell wall is thin. Right, that cell wall is thin, plus it is having more lipid, right? Outer lipid envelope will also be there, inner lipid envelope will also be there. So that is the main reason. So who's ready to take it as a challenge that ma'am, after your class today, I will read it from the chapter cell and I will definitely revise this topic? Yes. Who's ready to do that? So guys, the sessions are not going to be, sessions are not going to be like this, that every day I will just teach, I will not ask you anything. No, right? The session should be interactive. No doubt, the proper interaction is missing on the YouTube platform. When you study in our batches, right, we can interact with you all. There is a kind of one-to-one connection which is missing on YouTube, but still, let's try to build it, right? Let's try to build it and let's get best out of these sessions. Okay? So whenever I say that yes, you have to do a homework, you should, okay, you should take it as a challenge that okay, she has given us something, we have to do it, and you can put the answer in the comment section. Fine. So who's ready to take it as a challenge that yes ma'am, after your session I will revise the gram positive and negative bacteria from the cell chapter? Yes, that I will revise it from the cell chapter. Okay, let's go then. Okay, so now let's come to the actual topic, that is kingdom monera. So we will also consider NCERT, we'll revise from NCERT. So bacteria, they are the sole members, they are the only members of kingdom monera. Any doubt? They are the only members of kingdom monera. Any doubt? Yes. Do you have any doubt here? I believe there is no doubt, right? It is the sole member of kingdom Monera. Am I right? Yes, so they are most abundant microorganism. They are present everywhere. So when we say, see, they are saying they are present almost everywhere. So can I say that cosmopolitan word? Can I use this word cosmopolitan in distribution? Cosmopolitan in distribution. Can I use this word here? If something is present everywhere, we use the word cosmopolitan. So cosmopolitan in distribution. Cosmopolitan in distribution. So they are present in handful of soil. They can live in the extreme habitats. What is the meaning of extreme habitats? Like let's say hot springs where temperature is very high. Okay, deserts, snow, deep oceans, and even you know that where salinity is too high, where acidity is too high, where temperature is very high. So bacteria can survive there as well. Okay, bacteria can survive there as well. Clear bache? Clear bache? And many of them live on other organisms as parasites. Right, as parasites, like some bacteria, they live inside our gut, right? They are helping us, right? That is one case. But some bacteria, they are dependent on other host organisms. They take their nourishment from them. That's why we are calling them as parasites. Okay, so they are basically grouped under 4 categories based on their shape. So yes, it is the another important point that you should know. Okay, that on the basis of shape itself, they are categorized. How are they categorized? These bacteria, they are categorized on the basis of their shape. So it is a very simple question, but again, it's a previous year question. It is a previous year question. So one is bacillus. Right, one word is what? It is bacillus. Then cocci, spirillum, and vibrio. Right, these are 4 different shapes. Okay, these are 4 different shapes. What's that? These are 4 different shapes. So when it is bacillus, it means it is rod shaped, means cylindrical. What is the meaning of it, bache? It means it is cylindrical. What's that? It is cylindrical. Cocci, it's plural, singular is coccus. Coccus, it means spherical. What's the meaning of this particular shape? The bacteria is spherical. Right, bache? The bacteria is spherical. Spirillum, the shape is spiral. The shape is spiral, and here it is comma shaped. Okay, vibrio is comma shaped. Okay, something like this. Here, okay, coccus, spherical. It can be diplococcus, spherical, and here rod shaped bacteria. Okay, rod shaped bacteria. Understood? Yes, right? So there are different examples there, and I believe you also know that examples, right? If you don't know it, don't worry, we'll discuss that too. Okay, we will discuss that too. So firstly, tell me the shape of the structure of bacteria clear? The structure of bacteria clear? Their cell wall, their cell coat, everything is clear. The genophore word, everything is clear. Yes? Sure. So we are talking about the shape of bacteria here. So students, if we read the things from NCERT, in the NCERT, they don't classify it properly. They don't mention it properly. Okay, they do not classify it properly. They do not mention it properly when we talk about the kingdom Monera here. Okay, so for your knowledge, I'm telling you, in the 6 kingdom classification system, we discuss it separately. It is given in NCERT but not explained properly. So kingdom Monera, it is subdivided. Right, actually this kingdom Monera, it is divided into, there are 2 sub-kingdoms. As per 5 kingdom classification system, there are 2 sub-kingdoms. So in one category, we consider Archaebacteria, right? Archaebacteria, and another is eubacteria. Another is eubacteria. So archae, here you are going to consider that primitive bacteria which can survive in the extreme conditions. Okay, Archaebacteria. Right, bache, Archaebacteria. And another category, right, another category is eubacteria. So eu means true. Okay, so this Archaebacteria can survive in extreme conditions. right? It lives in extreme conditions, right? In extreme conditions. So when we will discuss Archaebacteria, we'll talk about halophiles. We will talk about thermoacidophiles. We will talk about methanogens. Okay, that's what we are going to discuss here mainly. Is that clear? Is that clear? Is that clear, everyone? So can we recall what we have completed so far? Yes. Yes. Can we just recall what we have completed so far? Right? Tell me, students, do you want regular classes? Do you want regular classes? Do you want me to take the session with high energy? Right? So that we can finish the syllabus quickly. I believe you want that. If you want it, then show some hearts in the chat section. Quick. Show some hearts in the chat section. Quick. Yes. That yes, ma'am, we want that. So for that, you have to be energetic. You have to be consistent. You have to participate in the chat. You have to answer the questions. Okay, you have to answer the questions. Okay, so what we have discussed so far, we started with the basis of classification, with the basics, right? We talked about the 2 kingdom, 3 kingdom, 4 kingdom, 5 kingdom. Then we discussed viruses, virions, prions, lichens. Now we are talking about the kingdom monera. Now we are talking about the kingdom monera, having 2 subkingdoms as per 5 kingdom classification system. Archaebacteria, where primitive bacteria will be discussed that can survive in the extreme conditions, in the harsh conditions. So here we have halophiles, thermoacidophiles, methanogens. And when it is eubacteria, it's true bacteria. It's true bacteria. Okay, it is true bacteria. Right, bache? So here you will discuss bacteria, mycoplasma. Yes, what do we discuss? That simple bacteria, Mycoplasma, Cyanobacteria. Basically, you are dividing bacteria into different categories. Okay, Actinomycetes. So in NCERT, as per your syllabus, we have to focus on these 3 mainly. We have to focus on these 3 mainly. So when it is Archaebacteria, halophile, thermoacidophile, methanogens, and when it is eubacteria, so it is having bacteria, mycoplasma, cyanobacteria, actinomycetes. Is that clear? Is that clear? So now the next to it is the classification of bacteria as per its shape. So here you are talking about the eubacteria. What are we discussing here mainly? We are talking about the eubacteria first. So we started with the shape part. Is that clear? We started with the shape part. So bache, we also discussed the flagellation. Right? We also discussed the flagellation. Right? I'll not go beyond NCERT. Right? Only the PYQs I'm going to consider. Only the PYQs I'm going to consider. Rest you have to focus on NCERT only. So as per flagellation, so depending upon absence or presence of flagella and their number, we have to divide bacteria further. Okay, so if a flagella is present, it means the bacteria is mobile. Yes, ma'am. So bache, flagella here is going to be very simple kind of flagella, and that is made up of flagellin protein. Right? In the case of monera, flagella is not made up of microtubules. It is made up of flagellin. It is made up of which protein? It is made up of flagellin. It is made up of flagellin. So that's simple, right? Simple basic body, simple hook, and that thread will be there. A very simple structure of flagella will be there. Okay? Okay. So if flagella is absent, then the word will be atrichous. That flagella is not there. Now, if flagella is present and there is only one flagella, monotrichous, only one flagella is present. Only one flagella is present. So that is how we have different words. Monotrichous, right? Amphitrichous, cephalotrichous, like this. Okay, like this. Is that clear? Is that clear? So you are already aware of its structure, am I right? You already know about its structure, am I right? Yes. So now the next topic that we are going to discuss here is the reproduction in the case of bacteria. I'll share the flow chart with you all as well. Okay, so what is next? Reproduction in bacteria. So what do you think in bacteria? What kind of reproduction is there? Let's complete it, students, and then we'll focus on the NCERT. Don't worry. Right, the nutrition and the reproduction part, it is important, and the difference in, right, and the difference in your Archaebacteria and eubacteria. So you tell me, what do you know about this? See the shapes. This diagram is from NCERT. The shape, you can see spherical, bacillus. This is showing the endospore, the spirillum, and comma shaped. Right? So that's what we have to cover. Okay, so what I just said, that we have to discuss the reproduction in bacteria now. What we have to discuss? Reproduction. So what kind of reproduction will be there? Sexual or asexual? Yes? Sexual or asexual? Guys, question is, what kind of reproduction is there? Sexual or asexual? Mainly sexual is there. Students, see, it's a primitive cell. Mainly you are going to talk about the asexual reproduction. If you think that proper sexual reproduction will be there, no, it's not like that. Okay, you, in the case of bacteria, you won't see that they are going to form the gametes. Right? You're not going to see that they are going to form the gametes. Okay, the gametes are going to fuse. No doubt, it's correct that from one bacteria, genetic material will be transferred to another bacteria by different ways. So sometimes we say that it's a kind of sexual reproduction, but proper sexual reproduction where gamete formation is there and all, that is not there in the case of bacteria. Okay, so mainly you will talk about yes. So what are we going to discuss here? We will talk about the binary fission and sporulation, means spore formation, and there will be genetic recombination. These are the three things. There will be genetic recombination, binary fission, sporulation, genetic recombination. Binary fission, sporulation, genetic recombination. That's what we have. These are the three ways by which the bacteria will divide. Okay, by which bacteria will divide. So the very first is binary fission. So let's talk about it. So what do you understand by this word, binary fission? What do you understand by this word, binary fission? Binary, bi means two, fission. Right, like let's say one cell will divide into two, simple. One cell will divide into two. This is the simple multiplication. Okay, this is a simple multiplication. It is the most common in all kinds of bacteria. This binary fission, it is most common in all types of bacteria. What's that? It is most common in all types of bacteria. What is it? It is your binary fission. It is your binary fission. Is that clear? Is that clear? So, how is it going to happen? See, when the conditions are favorable. Okay. So, under favorable conditions. Right. What is the meaning of favorable conditions, students? Yes, people, tell me quickly. What is the meaning of favorable conditions? We are saying under favorable conditions, right? What are we saying? We are saying under favorable conditions. So what is the meaning of favorable conditions? See, when proper temperature, right, the pH, everything is perfect, right? When everything is perfect for that bacterial growth, right? Sometimes let's say if the conditions are not favorable, right, the outside environment, it is not favorable for bacteria, then it will not divide. So when the conditions, they are favorable, right, they are favorable. Is that clear? Is that clear? So bacterial cell expand. Okay, bacterial cell expands. What will this cell do? It is going to expand. DNA, then DNA replicates. So here in the case of bacteria, there is nothing like mitosis, meiosis, no. Normally in bacterial cell, it will expand, its DNA will replicate, it will attach itself to mesosome. So do you remember in the last class also when we were talking about the mesosome? These are the finger-like enfoldings in bacteria. They are having the respiratory enzymes, right? Plus they will help in dividing the DNA equally to the two daughter cells. Do you know that? Yes. Do you remember that? Do you remember that? Tell me, do you remember that? Yes. So basically bacterial cell will expand. I'll show you the diagram. Right. So here you guys can see. Can you see that bacterial cell is, yes. Can you see that bacterial cell, it is growing. Right. It is expanding. The DNA is replicating. Can you see that? The DNA is replicating. Yes. This DNA will attaches itself to mesosome. A new mesosome will also form. Right. A new mesosome will also form. And then basically this DNA, can you see that? It is kind of dividing, right? Right, because it has replicated already, 2 copies are there, it is attached to mesosome, a new mesosome will form, and slowly what will happen? This constriction will start, and then finally the cytoplasmic division, and you will get the 2 daughter cells. Is that clear? Is that clear? So cytoplasm will also undergo a centripetal constriction. Can you see that? Yes. So basically the cell is dividing. Cytoplasm is also undergoing the constriction. So it is from periphery towards the center. From periphery towards the center. So that is why I am using the word centripetal constriction here. Okay, centripetal constriction here. So that is happening and the cells will divide into 2. This is the binary fission. Is that clear? Yes, bache. Is that clear? This is about the bacteria. This is the binary fission. Is that clear? So there is one line that you should know, that the time taken by this cell, okay, the time taken by cell, can you see that? The time taken yes, so the time taken by the cell to divide into 2 is known as the generation time. Okay, so students, again I'll request you, this is a very simple chapter. Okay, I'll say 3 to 4 questions will come from this chapter. You can get 1 question from fungi, 1 question from bacteria, but these things are basic. What is generation time? How a cell is dividing? Is it having mitosis, meiosis in bacteria? So that things are also related with the prokaryotic cells. So you better pay attention here, okay? Okay, so I'm writing it here, right? The time taken by bacterial cell to reproduce is called generation time. What's that? It is known as generation time. I'll give you one example which is there in NCERT. In the case of E. coli, Escherichia coli, the world famous bacteria. If you are a NEET aspirant, you should know about E. coli. You should know about E. coli. You cannot leave E. coli. E. coli, it's a gram-negative bacteria. It's a gram-negative bacteria. So this E. coli, okay, its generation time is 20 minutes, right? Yes, its generation time is 20 minutes. In 20 minutes, it will divide, right? It will divide into 2. So basically in a bacterial cell, you people will see that there is the circular DNA. What kind of DNA is there? There is the circular DNA. Okay, so in both the direction, the replication is there. That's why in E. coli, it is for 20 minutes. This point clear? This point clear? Yes? Okay. Right? Okay. Is that clear? Any doubt? Any doubt? Because next is sporulation. So here actually what is happening? This is binary fission. Multiplication is going on. Can I say so, students? Multiplication is going on. Can I say so? Ultimately, what is happening here? Multiplication now. One cell is dividing into two. That two cells will divide into four. Four cells will divide into eight. Multiplication. So the next division is sporulation here in bacteria. Right. The next division is sporulation. So here also bacteria will increase, but without multiplication. Here bacteria is not going to multiply, but still we'll see the number will increase. So let's discuss that. So what do you understand by the word sporulation? Right, sporulation means spore production. What is the meaning of sporulation here? What is the meaning of sporulation here? Spore production. Right, spore production it is. What is it, students? What is it, students? It is the spore production. It is the spore production. So bacteria, these bacteria, they produce some spores. If they are formed inside, endospore, right? Endospore. So bacteria, they produce endospore. What's the word, bache? Endospore. And how will they produce endospore? In response to adverse condition. In response to adverse conditions. Okay, like let's say for you, if things are favorable, you have sufficient time, you are studying properly, obviously you can complete your syllabus on your time, you can give paper, you can give your NEET paper with your full efficiency and you can get good marks, isn't it? Let's say if conditions are worse for you, your ways will change. Let's say you didn't get the sufficient time, you got sick or some family problem is there. So accordingly you will complete your syllabus. Accordingly you are going to attempt the paper now. So same way for bacteria, if environmental conditions, they are favorable, they'll go for binary fission, they'll multiply. Let's say conditions are not favorable, right? That temperature or the surrounding, it is not in the favor of that division or something. Bacteria can even die. So in that case, when the conditions are adverse, when they are not favorable, the bacteria, they form the spore. Right? What will they do? They are going to form the spore and we are calling it as the endospore. Right, bache? Right? So endo means inside. So they'll produce inside the bacterial cell. Yes? 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 Right? They'll produce inside the bacterial cell. So endospore. So this endospore production, right? Because endospore will form inside the bacteria. So this endospore production can also help bacteria to survive in unfavorable conditions. Am I right? To survive in unfavorable conditions. Yes, ma'am. Definitely this bacteria is helping in that way. The spore production. Let's say, I'll give you the example. Let's say here you have the bacteria. Okay, the spore is produced inside. So when conditions are favorable, it will stay like that. Unfavorable, it will stay like that. Let's say now conditions are favorable. The cell wall will burst. The spore will come out. Again, it will grow like a bacteria. Again, it will grow like a bacteria. Isn't it? Again, it will grow like a bacteria. So can I say that it is a method of perennation? Yes, in the morphology we use this word, method of perennation, means something which is helping that particular part to survive in the unfavorable conditions. Yes, like in the case of plants, we discussed now that they are having even the rhizome, ginger, you know that can survive in the unfavorable conditions. When the conditions are favorable, from that a new plant can form. So that is the point. Okay, that is the point here. Is that clear? Is that clear? So these endosperms, they are highly resistant to desiccation, temperature and chemicals. Okay, these endosperms, they are highly resistant. Okay, they are highly resistant to desiccation, temperature and chemicals. So that is how it will help the bacteria to survive in unfavorable conditions. Okay. Okay. Right? Yes. So I'll give you one more example. Bacillus. It is pathogenic. Okay. So Bacillus shows the endospore production, endospore formation. Right? This Bacillus, it shows endosperm formation. In the chapter biotechnology, we discussed this. You remember now, Bacillus thuringiensis. Anyone in the class? I believe droppers are also watching this lecture. Right, so Bacillus thuringiensis, we discussed it is going to form the endospore. What is that endospore? It is a kind of toxin, right, that can go inside the insect and that can destroy its midgut epithelial, and that insect can die due to starvation. Do you remember that? So Bacillus and Clostridium, these two pathogenic bacteria, they actually form these spores. They actually form these spores. So do you remember that? Yes, students, do you remember that Bacillus thuringiensis? It is having the endospore, right? We say it forms a protein crystal. We call that as prototoxin, right? So that is the same thing. That's how we are relating these two things. Okay, so this part clear, students? This part clear? So next is the genetic recombination. Okay, next is the genetic recombination, which is also known as reproduction, kind of reproduction in bacteria, and it is also known as, yes, genetic recombination, right, which is basically what? Basically the sexual reproduction or parasexuality. That is the word that we use here. So genetic recombination, kind of sexual reproduction in bacteria. So 3 words are there that we have to discuss, and yes, this is important. Okay, this is important. So up to this part all clear, yes or no? Up to this part all clear, yes or no? Tell me, students. Yella, Selvi, Sukhdev, Pooja, is it clear or not? Or do you have any doubt? Yes, or do you have any doubt? Done? Okay, so let's start. So we are going to discuss 3 things here. Okay, we are going to discuss 3 things here. So basically it is going to be, yes, transformation. All these 3 words are important. It is going to be transduction and it is going to be conjugation. In the biology, while reading the chapters like molecular basis of inheritance, biotechnology, we'll use these words. Okay, transformation, transduction, conjugation. Transformation, transduction, conjugation. Again, repeat it with me. Transformation, transduction, conjugation. Clear, bache? Clear, bache? So it is the kind of sexual reproduction. So it is not true sexual reproduction. If you think it's a true sexual reproduction, it is not there. Why? Why? Because there is no syngamy in bacteria, no syngamy, no meiosis. Right? Because there is no syngamy and no meiosis in bacteria. That's why we are saying that it is not the true sexual reproduction. Okay, that is why we are saying that it is not the true sexual reproduction. So the first thing is transformation here. Let's talk about it. So can you quote any example here in the transformation part? Any example? Yes. I'll give you a chocolate if you will answer it correctly. I'll give you a chocolate if you will answer it correctly. Quick. Quick. Any example of transformation? Even this word is explaining it. Okay, even this word is explaining it. Transformation. Yes. Transformation. This word is itself explaining what's going to happen. Transformation. To transform. Let's say you are transforming one bacterial cell into another. You are converting one bacterial cell into another. That is the transformation. That is the transformation. Are you getting my point? Let me explain. Let's say there is a bacterial cell. There is a bacterial cell. Or there is a genetic material in the surrounding. There is some genetic material, a piece of genetic material, a piece of DNA is there in the surrounding. If this bacterial cell, if it picks this, if it will pick this genetic material from surrounding, if it incorporates this genetic material in its own DNA, right? Just as this, see, this is the genetic material outside this bacterial cell in its environment. It has picked it up. Okay, now this cell, this genetic material is the part of this bacterial's DNA. Right, it is the part of this bacteria's DNA. So obviously, whenever this bacteria's DNA will divide, basically it will do transcription. It will form mRNA, then protein. So in simple words, right, because this outside genetic material, it is also, right, it is also incorporated here in this bacteria's DNA. So definitely this DNA will also express itself. This DNA will also express itself. It will also make proteins. Okay, it will also make proteins. Okay, so now if, because this bacteria cell has picked up some genetic material from the outside, it will also have that genes. So that is going to transform this bacteria. Na? It is having the new genes, isn't it? It is having the new genes. So here what is happening? Bacteria is absorbing DNA from either from its dead relatives or, right, let me write it here. Bacteria absorb DNA either from its dead relatives or from outside environment or from outside environment. That is what transformation is. This bacterial cell is also transformed, right? So where do we study it? Even in your book, in your NCERT, you can write it. Where do we study it? We study it in the molecular basis of inheritance, Griffith's transformation experiment or transformation principle. There you will discuss it in detail. Okay, Griffith's transformation principle. You will study it in molecular basis of inheritance, and there we'll discuss it in detail. Is that clear? Is that clear? Yes. So next is the transduction that we will study, and then we'll talk about the conjugation. Okay. After this respiration part and nutrition, then kingdom monera will be finished like this. Then we'll move to the protista. Right. So transformation. So next is what? Transduction. Again, a very commonly used method. Again, in genetic engineering, you will study this transduction. Okay. So, yes, do you remember anything related to the bacteriophage? Yes, do you remember anything related to the bacteriophage? Bacteriophage, we have discussed the bacteriophage. If you remember this diagram, polyhedral like this, double-stranded DNA, if you remember, this bacteriophage. What is this bacteriophage? It is a virus. It's a DNA virus. And it is a virus that infects bacteria. It is a virus that infects bacteria. Bacteriophage. It is a virus that infects bacteria. That is a bacteriophage. What is it? It is a bacteriophage. So it's a virus on a bacteria. Right? It's a virus that is eating a bacteria. It's a virus basically that is going to, it's a virus that is parasitic on a bacteria. Bacteria is itself parasitic, and now a virus is parasitic on a parasite. So we will also call it as hyperparasite. Right? We can also use the word hyperparasite for it. Okay? Hyperparasite for it. Right? So when a parasite is parasitic on a parasitic host, right? When a parasite or when a virus is parasite on a parasitic host, you can use the word hyperparasite for that, right? So this is bacteriophage. This is bacteriophage. It's a virus that can infect the yes, bacteria. So what's the point here? What is the point here? So here the point is very simple, right? The transfer of genetic material from one bacteria to another. What is transduction? It is the transfer of genetic material from one bacterium is singular of course, to another, from one bacterium to another through a virus. That virus is bacteriophage. So when you use a virus to transfer the DNA from one bacterium to another, you use the word transduction. I will give you example here. Let's say this is one bacterial cell. This bacteriophage is going to infect it. Okay, this bacteriophage is going to transfer its DNA into this bacteria. This DNA will also get, right, this bacteria virus DNA will enter in the bacterial cell. So you know that this DNA will use the host machinery. Am I right? This DNA, it is going to use the host machinery. It is going to use this bacteria's machinery, and it will divide. So within bacteria, you will see that there will be multiple copies of this bacteriophage, right? There will be the copies of this bacteriophage DNA. So actually that bacteriophage DNA will also, right, because it is using the host machinery, it is also incorporated with the host DNA. So obviously when these bacteriophages, then they will start infecting another bacterial cells. They are transferring the genetic material of this bacteria into the another bacteria. Are you able to understand it, guys? Are you able to understand it? Tell me quickly. Are you able to understand it? This is what transduction is. This is what transduction is. Okay, so it's the transfer of genetic material from one bacterium to another through the virus. Okay, through the virus. This is the transduction. What's that? It is the transduction. Is that clear? So yes, even in molecular basis of inheritance, there is another experiment that we study, right? So I'm giving you the example here. You can note it down in the NCERT. That is Hershey and Chase transduction experiment. Okay, a blending experiment. So what is transduction? When virus is used to transfer the genetic material from one bacterium to another. So when students read such chapters in class 12th and when teachers ask, do you know what transformation is? Do you know what transduction is? Do you know what conjugation is? They are not aware of it, right? They are not aware of it. They don't know what these words are. Now I believe it is clear. Now believe these meanings are clear. So guys, see, reading NCERT doesn't mean, right, that you are just reading it line to line and then you are like, okay, I can translate one line. You have to understand it, right? I emphasize this thing a lot, right? Reading NCERT doesn't mean that you have to just translate the lines. You need to understand that lines. You need to understand the each and every word given in NCERT. You need to decode the NCERT, okay? And that is why sometimes we have to relate one chapter with another. Sometimes we have to mix the topics. Then only we can understand the overall subject biology. Okay, so even if you are still confused that how to read the NCERT, I think tomorrow itself I posted one video. So this is the video in which, right, I have discussed that how can you read the NCERT properly, what are the things that you need to take care, right, the points that you need to make, okay, the way you need to make the notes. Okay, so everything is discussed in this video. So after the class, you guys can check it. Okay, after the class, you people can check it. Is that clear? So transformation, transduction, next is the conjugation. Next is the conjugation. So do you know what is conjugation? What is conjugation? Yes. Do you know what is conjugation? Tell me, what is conjugation? See, let's say there is a bacteria. This is one bacteria. That is the nucleoid of bacteria. You remember nucleoid, devoid of histone proteins. We also use the word genophore for that. We also use the word genophore for that. So this is your one bacteria. Now this bacteria is having a fertility factor. Okay, let's say it is having a plasmid. It is the F plasmid. What is the meaning of this F? Fertility. Fertility. So actually some bacteria, they are having this F plasmid. It is having the F factor. It is having this F factor. So F stands for fertility. F stands for fertility. Such bacteria are even considered as F positive. We also call them as F positive bacteria. Understood? They are having the fertility factor. Okay? Okay. So now let's say there is another bacteria. This is another bacteria. It doesn't have no F factor. No F factor. Right? It is without F factor. So this F positive bacteria, this is F negative bacteria because it doesn't have that F factor. So F factor is the fertility factor. So it is believed that this particular bacteria is considered as a male, or you can consider it as a donor. And this bacteria is considered as female, and you can consider it as a recipient. Okay, recipient. Now this particular bacteria will transfer its, let me draw that too. This particular bacteria is going to transfer its F factor to another bacteria. How? It will make a tube-like structure. How? It will make a tube-like structure. What will it do, students? It is going to make a tube-like structure. What is this tube? Right, this is the conjugation tube. This is the conjugation tube. And this bacteria can form it because it is having the F factor. So what tube it is? It is the conjugation tube. Right? It is the conjugation tube. Is that clear? Is that clear? Right? So this conjugation tube will form. It is also known as pilus. In the bacteria, in the chapter cell, we discussed now pilus. This word sex pilus, and plural is pili, sex pili, which is made up of pilin protein. So it's a tube-like structure. It's a tube-like structure, right? It will form, and now next is what? This tube-like structure will join with this another bacteria, and this F plasmid, its genetic material will be transferred to the another bacteria. Can you see that? The genetic material will be transferred to the another bacteria. And finally, what are we going to see? We'll see that this bacteria is also having the F factor. Now this bacteria will also contain this F factor. It's like that. Okay, it is like that. So conjugation tube will bring these two cells together. Okay, two cells together. A part of genetic material, a part of this plasmid is transferred here, and it will result in the formation of the F plasmid there. So this is the conjugation. That is how the genetic transfer will be there. Is that clear? So this, right, bache? So now both will become F positive, F positive. Okay, both will become F positive, F positive. Okay. Yes. Yes. Doubt? Any doubt? So can we revise it from NCERT? Can we revise it from NCERT? Yes, can we revise it from NCERT? Then let's go. Okay, so bacteria are grouped under 4 categories. We have already discussed coccus, bacillus, vibrio. Vibrio is singular, vibrio is plural. Spiral, spirillum is singular, spirilla is plural. Okay. Okay, so we are talking about the eubacteria. So bacteria structure is very simple, but they are having very complex behavior. So compared to many other organisms, they are the group which show the most extensive metabolic diversity. Means in their metabolism diversity is there. If you compare one bacteria, right, the same kind of bacteria, you will see the metabolic diversities, right, bache, you will see the metabolic diversity. So some of the bacteria, they are autotrophic. They can make their own food, right? They may be photosynthetic autotrophs or chemosynthetic, and some are heterotrophs. So now next portion that we have to discuss is the nutrition. But before that, let's discuss the respiration, and then we'll finish these cyanobacteria and all, and then we'll come to the archaebacteria thing, right, students? Okay, let's go. So another important words that we have to use here is aerobic bacteria and anaerobic bacteria, right? Respiration. So obviously, if you will attend the classes, if you will understand the topic, right, in that depth, then NCERT reading and all the questions, That can come from NCERT. That will be easy for you, isn't it? Okay, so you know that we will have two things. We'll talk about aerobic bacteria and we are going to talk about the anaerobic bacteria. Aerobic and anaerobic. So what is the meaning of aerobic bacteria? What is the meaning of aerobic bacteria? Yes, students. The word here is aerobic. Yes, you know na, aerobic and anaerobic. So respiration is of two types, aerobic and anaerobic. Aerobic respiration and anaerobic respiration. So aerobic, in simple words I will write it. Aerobic, oxygen present, means oxygen is available. No oxygen is absent here. So some bacteria, they need oxygen for their respiration, some they do not need that. Okay, so here there are two words that you should know. One is obligate aerobes, another is facultative anaerobes. Now, obligate, even in the bacteria we have discussed it. Obligate, obsessed. What is the meaning of obligate? Means obsessed. Obsessed for oxygen. It needs oxygen. If there is no oxygen, this bacteria cannot survive. Okay, okay. So bacteria grow exclusively in the presence of bacteria. Okay, bacteria grow exclusively in the presence of oxygen. Oh, I'm sorry. Yes, in the presence of oxygen. It needs oxygen. Okay. Right, in the presence of oxygen, it needs oxygen. It cannot survive without oxygen. Okay, it cannot survive without oxygen. For an example, your azotobacter. I'm giving you one example, there are many. Now, facultative anaerobes. It is an aerobic bacteria. It is an aerobic bacteria, but can become anaerobic if oxygen is not available. It knows how to adjust. It knows how to adjust. Right? Right? Yes, bache. So these bacteria are normally aerobes, means normally they need oxygen, but can even survive without oxygen. Yes, students, but it can, yes, switch over to anaerobic conditions as well. So that's why it is facultative anaerobe. Right, that's why it is facultative anaerobe. So example, Clostridium. Again, this example is given in NCERT, Clostridium. Okay, so now if I am saying, I think now there is no need to explain it, you can understand it. If it is obligative anaerobe, what is the meaning of that? And what is the meaning of facultative aerobe? Yes. Do you know the meaning here? Do you know the meaning here? Do you know the meaning here? Obviously, obligate anaerobe, which needs the anaerobic conditions only. Facultative, it is normally, facultative aerobe, it is normally anaerobic, but can switch to aerobic method as well if the oxygen is available. Okay, if the oxygen is available. Is that clear? Yes, is that clear? So now let's move to the next part, that is the nutrition. That is the nutrition. Okay, so here we have this flow chart. One word is autotroph. What is the meaning of autotrophs? Autotrophic. They can synthesize their own food. They can synthesize their own food. They are capable of making their own food just like plants. Right? They are capable of making their own food just like plant. Autotrophs they are. So it can be chemosynthetic also. They can use the chemicals to make. They can oxidize different inorganic substance like nitrate, nitrite, and can use the food. This is one thing. This is one thing. And yes, some can be photosynthetic also. Photoautotrophs also. है ना? Photoautotrophs also. So in the case of bacteria, you will also see some are saprophytes. Means saprophytes are the one that can take their nutrition from dead and decaying organic matter, right? So they can derive their nutrition from dead remains. Okay, some are parasitic, they will take their nutrition from the host plant, and some are symbionts. They will live together they will show mutual relationship, mutual association, and can take the benefit out of it. So for the quick revision, that is what I have added here, right? From a book I have added this picture, right? So we'll revise it. So see, autotrophic bacteria, one is photoautotroph, another is chemoautotroph. Is that visible? Is it visible? Photo means light. These bacteria will use the light and make their own food, like your Cyanobacteria, blue-green algae. Okay, so here also they are having the categories. So you should know one thing, like purple sulfur bacteria is the example of photoautotroph. Green sulfur bacteria is the example of photoautotroph. So here from this flow chart only you can revise. I'll share the PDF in the Telegram group, and then you can revise with the help of that. Chemoautotrophs that will use chemicals, like purple non-sulfur bacteria, green non-sulfur bacteria. Okay, right? So they will use non-sulfur. Yes, so they will use non-sulfur organic compounds, and then they can make their food. Okay, students. And one is nitrifying bacteria. Right, nitrifying bacteria, iron bacteria, hydrogen bacteria, sulfur bacteria, they all are the example of chemoautotrophs. So when you talk about the evolution on this planet Earth, initially bacteria came. What kind of bacteria came? Heterotrophs kind of, not the autotrophs kind of. Initially heterotrophs came, and then with the time autotrophs evolved from heterotrophs. So remember this thing, right? So firstly heterotrophs came. So photosynthetic and this part clear, photosynthetic and chemotrophic clear, sulfur bacteria, purple sulfur bacteria and all. Sure. So heterotrophic may you know that, right? You know that they'll be needing what? The different source. So see, this is another flow chart for all of you. So that is just for an idea, right? So this is about the basic properties of the bacteria. Any doubt here? This is about the basic properties of the bacteria. Any doubt here? Any doubt here? So students, basically we are discussing eubacteria, right? So I told you there are the different categories also. So now we will talk about Mycoplasma. We are going to talk about Mycoplasma. Okay, that is what we have to discuss. Mycoplasma. Okay, so what do you know about Mycoplasma? What do you know about Mycoplasma? What do you know about Mycoplasma? It's a type of eubacteria. Yes? It is the organisms that completely lack a cell wall. Sorry. Completely lack a cell wall. It's the only bacteria which is not having the cell wall. It is not having the cell wall. Is that clear? Okay. Right, it is not having the cell wall, and it is mostly parasite. It is not having a cell wall. Okay, it is the smallest living organism. That is also a very famous question. It is the smallest living organism known. Right, and it is the smallest living organism that can survive without oxygen as well. Right, that can survive without oxygen as well. So can I say that it is facultative anaerobe? Can I say that it is facultative anaerobe? Tell me students, can I say that it is a facultative anaerobe? It can survive without oxygen. right? Though it is aerobic, but it can still survive without oxygen. So that's why we can use the word, it is facultative anaerobe. So it is given in NCERT, obviously important it is. Okay, so smallest living organism, remember, 0.3 micrometer. Mostly they are parasites, right? Mostly they are parasites. Okay, bache? Yes. So if you talk about the genetic material, that will be single linear double stranded molecule of DNA. It is single linear double stranded DNA molecule. That's it. So mycoplasma, mostly parasitic. So one thing, because it doesn't contain a cell wall, so obviously it doesn't have a proper shape. It doesn't have a proper shape. Can I say that? Because it is not having a proper cell wall, it simply means it doesn't have a proper shape, right? Because it lacks a cell wall. So that is why mycoplasma is also referred as jokers, jokers of microbiological park. We can also call it jokers of microbiological park, and along with that, jokers of plant kingdom, right? We can also call it as jokers of plant kingdom or jokers of microbiological park. Why? Because it is pleomorphic, right? It is pleomorphic. What is the meaning of pleomorphic? Means it is having different shapes. Why? Because it doesn't have a cell wall, right? It is having different shapes. It doesn't have a cell wall. So it is the jokers of plant kingdom. Jokers of plant kingdom. Why can we say that it is the joker of plant kingdom, not the kingdom monera? Because by that time, this mycoplasma, it was placed in plant kingdom. So that's why in some books it is also referred as jokers of plant kingdom. Okay, jokers of plant kingdom. Done, bache? Done. Okay, so this is one extra information for you. They are this mycoplasma, it is resistant to, do you know, it is resistant to penicillin. Yes, it is resistant to penicillin. It is an antibiotic, but this cannot kill the bacteria, and sensitive to tetracycline. Now let me explain. See, you know that there are antibiotics. Whenever we get the infection, there are different types of antibiotics that we can take. If there is any antibiotic that acts on the cell wall of the bacteria, there is any antibiotic that acts on the cell wall of bacteria, that is not going to kill mycoplasma. So penicillin, it acts on cell wall. It doesn't have cell wall. So penicillin cannot kill mycoplasma, but tetracycline can. Okay, tetracycline can. Is that clear? Is that clear? Right? So it is parasitic to animals as well as to plants. So I'll give you one example. Right? Mycoplasma pneumoniae. Yes, Mycoplasma pneumoniae. You know na? It can cause pneumonia to animals. Mycoplasma pneumoniae. So that's the one example that I'm giving you. Okay, and you know that there are many. Right? There are many. Okay, Mycobacterium tuberculosis is also there, that can cause TB. So this is mycoplasma, right? This is mycoplasma. Understood? Yes. So next category is, okay, let me see we can use it or not, Actinomycetes. So Actinomycetes is also a kind of eubacteria. Actinomycetes, no, it is also a eubacteria. It is also the example of eubacteria, Actinomycetes, right? Actinomycetes. Then we will talk about the Cyanobacteria. Okay, so what is this Actinomycetes? What is this Actinomycetes? It is basically, it is considered as the ray fungi. You can also call it as ray fungi. But it is considered as a connecting link. Imagine, it's a connecting link between bacteria and fungi. Imagine. In bacteria and fungi, there is a connecting link. Actinomycetes is considered as that connecting link. Imagine. Okay. So it's a group of unicellular. It's a group of unicellular. Means single cell. It's a group of what? It's a group of unicellular branched filamentous bacteria. branched filamentous bacteria. Okay, so it's a group of unicellular branched filamentous bacteria which resembles the fungal mycelia, which resembles fungal mycelia. Is that clear? Which resembles fungal mycelia. Okay, okay, so they are also kind of gram positive bacteria, saprophytic. It's gram positive, it's saprophytic, right, and facultative anaerobic. That's it. This information is sufficient for you. Okay, so that is also a type of bacteria. Is that clear? That is also a type of bacteria. Clear, bache? Clear, clear, clear? Yes? Okay, so next is the most important one, that is the cyanobacteria. Right, this is important. Cyanobacteria. It is also known as blue-green algae. Right, what is it? It is also known as blue-green algae, cyanobacteria. It is a photosynthetic bacteria, students. What kind of bacteria it is? It is a photosynthetic bacteria and oxygenic photosynthetic bacteria. Okay, some bacteria, they do the photosynthesis, but still they do not release the oxygen. That is non-oxygenic photosynthetic bacteria, like some bacteria that releases sulfur, right? So it is the photosynthetic bacteria. Okay, it is the photosynthetic bacteria, blue-green algae, cyanobacteria, cyanophysi. So it is the only bacteria which shows oxygenic photosynthesis. Only bacteria that shows oxygenic photosynthesis. Okay, so cyanobacteria, blue-green algae. You can also call it as cyanophysi. Okay, you can also call it as cyanophysi. Is that clear? Is that clear? So they are gram negative. You can find it everywhere, fresh water, marine water. Right, one second. I like. So they are also gram negative. Right, so you can found in fresh water, hot water, even in some hot springs also we can see them, right? So that is what? That is the cyanobacteria. That is the cyanobacteria. What is it? It is the cyanobacteria, photosynthetic bacteria. Understood? Yes, bache, understood? Okay, so they can be unicellular, colonial, filamentous, right? They can be aquatic, they can be terrestrial. Filamentous, can be aquatic, or terrestrial. So it's not mandatory that it will be aquatic only, it can be terrestrial as well. Okay, okay, done bache? Sure. So there is something very important related to this cyanobacteria. Okay, and that is they are always covered with a mucilaginous sheath. Please write down, it is always covered with a mucilaginous sheath. Mucilaginous sheath that can absorb water from the surrounding. Okay, that is something which can absorb water from the surrounding. Okay, so cyanobacteria always covered by a mucilaginous sheath. Done, bache? It is always covered by a mucilaginous sheath. So I'll give you one example as well. You all have seen that example. Right? You all know about this example. It is even given in NCERT. Yes, something like this. right? Something like this. Nostoc, it is an example of this, na? Nostoc. So this surrounding is having the mucilaginous sheath, right? It can absorb water, it can hold water as well. So if you will see, these are some cells. I'm naming it as heterocyst. I'm naming it as heterocyst. So students, actually, as I said, they are unicellular but can be filamentous. Means single cells, they can even join and they can form filaments, the thread-like structures like this. Okay, single cells, they can join and they can form the filamentous structures like this. Are you getting my point? They can form filamentous structures like this. Now, when you talk about these filaments, it's important, please pay attention. These filaments are of two types. It can be homocystous. It can be homocystous. It can be heterocystous. It can be homocystous. It can be heterocystous. Anyone in the class who can explain me the meaning of these two words? Anyone in the class who can explain me the meaning of these two words? I'm saying it can be homocystous. It can be heterocystous. Homocystous, hetero. Homo means same. Homo means same. Hetero means different, right? Hetero means different. If all the cells are of same type, all the cells are same, all the cells are same, then it is homocystous, right? Then it is homocystous. What is it? Homocystous. One type of photosynthetic cells are there, right? One type of photosynthetic cells are there, right? So the example is oscillatoria, right? The example is oscillatoria, I think, right? The example is oscillatoria. Lutoria, it's not A, it's O. It's O. Still, I'll check it. I think it's O. Oscillatoria, right? Now, heterocyst, right? If it contains different type of cells, you know the example is nostoc, even the Ambika, right? The example is nostoc. See this nostoc. So it is having some heterocysts also. It is having some heterocyst also. So even the nostoc, even the Ambika, they are the examples, though they are having the different type of cells. We are calling it as heterocyst. Now, what is the meaning of heterocyst? Yes, what is the meaning of heterocyst? That is important, right? What is the meaning of heterocyst here? Look at this nostoc structure. Overall, no doubt, it is a photosynthetic algae. It's a blue-green algae. It is photosynthetic. Now, here, these heterocysts, they are non-photosynthetic cells. Means in this particular blue-green algae, some cells are not photosynthetic, right? They are not going to do photosynthesis. This is heterocyst. Why? Because it is anaerobic. Oxygen is absent in these cells. Oxygen is absent in these cells. They are photosynthetic. This cell here is photosynthetic. So if in any algae, right? If in any cyanobacteria, some cells, different type of cells are there, right? Like some are photosynthetic, some are non-photosynthetic. We are calling it as heterocystus. Okay, so yes, you should know about the heterocyst. That is important, right? A note, you should write a short note on heterocyst, that what is a heterocyst. So one thing is clear, that it is a non-photosynthetic cell, but what is the importance of it, right? What is the importance of this heterocyst? Yes, can you tell me? Can you tell me? So I'm writing it here. Yes, it is non-photosynthetic. It is large sized, pale yellow. It is large sized, pale yellow. It is anaerobic. It is anaerobic. Very important. So because it is anaerobic, right? One thing is clear, it is not photosynthetic, but it can fix nitrogen. It's a nitrogen fixer cell. Yes, it's a nitrogen fixer cell. It can fix nitrogen. Okay, it can fix nitrogen. It is involved in nitrogen fixation. Okay, bache, it is involved in nitrogen fixation. Done. So this heterocyst, it lacks photosystem 2. So that's why 1 second. Heterocyst, non-photosynthetic, large size, pale yellow, anaerobic, fixed nitrogen. It lacks photosystem 2. Right now, if you don't know this, we will discuss it in the chapter photosynthesis. So right now you can include it in your notes. It lacks photosystem 2. So that's why it doesn't evolve oxygen. It doesn't evolve oxygen. So it contains an enzyme. It contains nitrogenase enzyme. Nitrogenase enzyme, and bache, that is the enzyme that fixes the nitrogen. Which enzyme is there in these heterocysts? Nitrogenase. Nitrogenase. So it contains nitrogenase enzyme that needs anaerobic condition. Okay, that needs anaerobic condition for fixation of nitrogen. So that is why it doesn't contain the oxygen. It is anaerobic. Understood? Understood? Sure. So that is the role of the cyanobacteria, and it is the first organism to evolve oxygenic photosynthesis. Okay, it is the first organism to evolve oxygenic photosynthesis. Understood? Yes. So here also, bache, you will see that in the case of these cyanobacteria, na, let's revise it from NCERT. This is the main point, especially the heterocyst point, right? Especially the heterocyst point. So do not miss it. Like see, here it is given na. This is your mucilaginous sheath. I told you it is always covered with a mucilaginous sheath. It can hold water. So heterocyst should be crystal clear to you. This is the diagram here. Okay, this is the diagram here. This is the filamentous nostoc. So obviously, because it can fix nitrogen, so thylakoid is also there, right? So thylakoids are present. You remember when I was teaching you bacteria structure, I told you like in some bacteria, some other membranous extensions are there. We call it as thylakoid, and that thylakoid will contain the pigment for the nitrogen fixation. Okay. Okay, so thylakoid is present there, that will contain, obviously without any doubt, the pigments for nitrogen fixation, like your chlorophyll A. Okay, like your chlorophyll A. Done, bache? Chlorophyll A, even beta carotenes, even phycobilins are present. Okay, chlorophyll A, beta carotenes, phycobilins, these pigments are present here in the thylakoids. Is that clear? So that should be clear to you. Done, bache? Yes? Any doubt? So reproduction can always be by, it can be by binary fission. Okay, bache? It can be by binary fission. Genetic recombination may occur. Okay? So that's it. So heterocyst is the most important point here. It is the heterocyst which is the most important point here. Done, bache? Sure? So there are examples of this blue-green algae which you should know. I'm writing it here only. Many examples are there, like your nostoc, anabina. They help in nitrogen fixation. There are other examples like spirulina. Do you know about spirulina? So it makes single cell protein. It is proteinaceous. Right, it is proteinaceous. It is used as a source of protein. Okay, spirulina, and even astronauts, they carry it to the space, right? Because it's a very good source of food. Is that clear? It's a very good source of food. So, done, bache? It is a very good source of protein. Very good source of protein. So that's why you have to remember this. Fine, you have to remember it. It is important. It is important. Right? Spirulina. It is protein rich. So astronauts, they carry it. It's a previous year question. Astronauts, they carry it to the space with them. Fine. So see this bacterium dividing, this is, okay, chemosynthetic. So see in the NCERT itself, they have used the word cyanobacteria. We have discussed it. Right, this is photosynthetic autotrophs. It will do photosynthesis. Heterocyst, it is discussed. Gelatinous sheath is given. Okay, chalo, I'll help you to revise it. So there are thousands of different eubacteria or true bacteria, right? They are having rigid cell wall. So you know that that cell wall is made up of, peptidoglycans. Right, that cell wall is made up of peptidoglycans, NAM, NAC, murines they are. Understood? And if bacteria is motile, flagellum. If flagella absent, if a flagella is absent, you use the word atrichous. Right, you use the word atrichous. Then cyanobacteria, blue-green algae, it is having chlorophyll A? Yes, ma'am. Like green plants, and it is photosynthetic autotrophs. So they can make their own food with the help of light. Okay, so they are unicellular, colonial, filamentous. They can live in fresh water, marine water. They can be terrestrial also. So colonies, they are generally surrounded by gelatinous sheath, gel like it is. So we also call it as mucilaginous. It can hold water. It can hold water. So these, they can even form blooms in the polluted water bodies. So some of these organisms can fix atmospheric nitrogen because they have heterocyst. Okay, so nostoc is the example, Anamina is the example. Is that clear? Rest, chemosynthetic autotrophic bacteria, they can oxidize various inorganic substances, nitrate, nitrite, ammonia, and they can use the energy for the ATP production. We have also discussed this. So it helps in nitrogen, phosphorus, iron and sulfur recycling. So that flow chart will help you to revise this part. Then heterotrophic bacteria, most abundant bache, they are decomposers. Okay, so they are helpful in making curd from milk. If you remember, they are talking about the lactobacillus here. Lactic acid bacteria that can form the curd. Okay, some can form the antibiotics also. Some can fix nitrogen in, right, legume roots. Remember, rhizobium. So examples should be there on your fingertips. Right, root nodules of leguminous plants, they are having this rhizobium. Okay, so rhizobium when lives freely, when free living, then it doesn't fix, then it is aerobic also. Right, and doesn't fix nitrogen, and when it lives in symbiotic association, then it is anaerobic and fix nitrogen. Okay, so from these lines, that's how you have to decode the NCERT. Some are pathogenic also. There are many pathogenic bacteria to human beings, crops, farm animals and pets. So one bacteria that can cause infection to human being, Mycoplasma is also the example, right? There are other bacteria also. Can you give me any example of the pathogenic bacteria? Yes, can you give me any example of the pathogenic bacteria? There are many. There are many, right? Many bacterial diseases that we know. Salmonella typhi, Salmonella paratyphi, botulism, food poisoning is caused by bacteria. So there are many examples. So see here in the NCERT itself, they have given this cholera, Vibrio cholerae, typhoid, Salmonella typhi, tetanus, Clostridium tetani, citrus canker. It's in the case of plants. It is in the case of plants. So they are the disease caused by different bacteria. So that is your homework, students. That is your homework. Okay, so you have to mention their causative agents in the chat section. Okay, in the chat section, mention their causative agents. Right, the citrus canker and everything. Citrus canker, it's very common in the case of plants. Okay, so see, they divide by fission, discussed. Spore formation, discussed. Sexual reproduction, discussed. Mycoplasma, already discussed. Right, so that's how we are also covering the NCERT in our lectures. Don't you think so? That is how we are covering the NCERT in our lectures. So the next thing that we need to discuss is the Archaebacteria. Okay, the next thing that we need to discuss is Archaebacteria. Ready, students? So Archaebacteria that can survive in adverse conditions. So the first question arises here is why? Why Archaebacteria, they can survive in adverse conditions? Why can they survive in adverse conditions? That is the first question that we have. Yes, that is the first question that we have, that why Archaebacteria is capable of surviving in the adverse conditions. Yes, anyone? Yes, anyone? Anyone in the class? Archaebacteria. So one thing is, bache, they are the ancient bacteria. Isn't it? They are very old. So when conditions were hostile, na, then they were present. Okay, then they were present. Imagine when conditions were hostile. So they are the ancient bacteria that evolved when conditions were hostile. Extreme conditions were there. Then they evolved. These Archaebacteria. Okay. Now, they do not contain, that's a very important point, peptidoglycan. They do not have peptidoglycan. Do you know that? They do not have peptidoglycan. They do not contain peptidoglycan and cellulose in their cell wall. They don't have these two things. Do you know that? They don't have these two things. Yes. So they do not contain peptidoglycan and cellulose in their cell wall. So cell wall is made up of non-cellulosic polysaccharide. Okay, cell wall is made up of, so that's a very important difference. Cell wall is made up of non-cellulosic polysaccharide, doesn't contain these murines. Okay, and that is why, because their cell wall structure is different. It is also one of the reasons that they can survive in the extreme habitats, but eubacteria cannot, right? But eubacteria cannot. Even there is a difference in their cell membrane as well. Okay, so cell membrane is having monolayer of lipids. Yes. Right, so in the cell membrane, these differences are important, students. So in the cell membrane, what will you see? You will see monolayer of lipids. You will see single layer of lipids to which branched fatty acids are attached. That decreases the fluidity. So in their case, because fluidity is less, okay, that's why they can also survive in extreme conditions. So they are the important points, right? So they are having the branched fatty acid chains. Okay, bache? Okay, and there is some 16S ribosomal sequence which is different from another. That is also the point. Okay, so again I have added the differences here in the form of this. So see this, Archaebacteria and bacteria. So Archaebacteria mein ribosome present? Yes, ma'am. Cell wall is made up of pseudopeptidoglycan. So non-cellulose polysaccharide and even peptidoglycans are absent. Now, habitat is extreme. Okay, and other than that, these are some other differences, right? Like protein synthesis mein, in Archaebacteria it is the methionine which is the start codon, but in the case of normal bacteria it is formyl methionine. Okay, so that are the differences here. And there is one more difference, you can see this. So in Archaebacteria, cell membrane, no doubt it is having monolayer. It is having the monolayer of the branched fatty acids plus ether linked lipids are there. And here in eubacteria and bacteria, ester linked lipids are there which are attached with the proteins. So in eukaryotes, there are ester linked lipids which are attached to the protein. There are ester linked lipids which are attached with this D-glycerol, obviously, right? And here ether linked that are attached with L-glycerol. These are the differences. And because of that, Archaebacteria can survive in extreme conditions. Okay, bache? Okay, bache? So see, in eukaryotes, histone proteins are there. In eubacteria, it is not there. But in Archaebacteria, histones, similar to histones, some proteins are present. Okay? Okay, so that is the difference. You can take the screenshot and I'll share the PDF in the Telegram group as well. So this is about these Archaebacteria. Why are they different, right? Why are they different from the eubacteria? So examples are important here, right? See, the one that can survive in salty areas, they are halophiles. The one that can survive in salty areas, they are the halophiles. What are they? They are the halophiles. They can survive in saline conditions. Okay, bache? Okay, done. So halobacterium is the example. Halobacterium is the example. It's a very simple example. Then hot springs, thermoacidophile. So thermoacidophile is the word. Thermal word is associated with the temperature. Acid means the acidity is less. So they are in hot silver springs. Right? We can see them in the hot silver springs as well. Then methanogens bache. Methanogens bache that can survive in marshy areas. So they are what? They are anaerobic. They don't need oxygen. They are anaerobic. Okay, bache, they produce methane. Right? They produce methane. Right? So it is given here. Methanogens are present in the gut of several ruminants like buffalo, cows, and they are responsible for the production of methane. Right? So during the biogas production, that's what we discussed. So you can keep it in your mind. These methanogens, they are anaerobic. What are they? They are anaerobic. Is that clear? Is that clear? So this is all about, right? This is all about the kingdom monera. So this is the diagram of a bacterial cell. Hope it is clear to you all. Right? Hope it is clear to you all. Right, students? So they see halophiles, okay, halophiles, thermoacidophiles, and methanogens. Right? So the next is kingdom protista. Okay? Next is kingdom protista. So who gave it first? Ernst Haeckel, right? He introduced it first. Right? So it contains what? Unicellular eukaryotic cells. Unicellular eukaryotic cells. So kingdom protista. It is kingdom protista. So what are we going to study here in kingdom protista? It is CDESP. Please repeat it. It is CDESP. It is CDESP. That's the trick we have for this. It is CDESP. 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 So what is that CDESP? C stands for chrysophytes. Right? D stands for, so in this sequence itself you have to remember this, dinoflagellates. D stands for dinoflagellates. E stands for euglenoids. Okay, E stands for euglenoids. S stands for slime molds. And then there is P, that stands for protozoa. That stands for protozoa. So kingdom is protista. The trick is C, D, E, S, P. Right? CDESP. So please write the CDESP in the chat section quick. Quickly do it. Yes? Do it. It's CDESP. Chrysophyte, dinoflagellate, euglenoid, slime mold, protozoa. Chrysophyte, dinoflagellate, euglenoid, slime mold, protozoa. Chrysophyte, dinoflagellate, euglenoid, slime mold, protozoa. So that is what we have in the kingdom protista. Okay, that is what we have in the kingdom protista. Is that clear? CDESP, CDESP, CDESP. Right? So chrysophyte, here you will discuss about the diatoms and desmids. Okay, then dinoflagellates are there. Right? Then euglenoids, then slime mold, like this. Okay, so let's complete this. One second. Okay, so students, let's complete it tomorrow because at 2 p.m. I have another class, so I cannot complete it in one stretch. So for tomorrow, otherwise only fungi will be left. So let's complete protista and fungi tomorrow. It will be done in 2 hours. Right? It will be done in 2 hours. And let me tell you, from fungi, you guys can expect 2 questions individually from the fungi itself. And let's keep one session on Sunday, not on Saturday. I'm going to keep that session on Sunday. Right? Sunday. Still, I'll update you people. I'll tell you tomorrow. So there will be the 5 years PYQ practice of this chapter, biological classification. We'll practice the last 5 years questions, right? Or 5 to 10 years. Let's keep it 10 years. That will be better. Last 10 years PYQ. What say? Last 10 years PYQ from the chapter biological classification. So from the next week, let's start plant kingdom and we'll finish that in 3 parts again. Then animal kingdom will be there and that's how we are going to finish the diversity part. So I'll keep one lecture where we'll finish the living board. Okay, so after the class I will share the PDF in the Telegram group, and students, I'll suggest you people to revise it thoroughly from the NCERT and practice as much questions as you can. So when I will keep the session, the practice session on Sunday, you will get an idea right about the revision thing also. Okay, so do not forget that from today itself they have started this offer. Okay, they have started the offer, that is you can get the 100% cash back and free flashcards. Okay, so enroll in the batch today. In the description box of this particular video there is a link, you have to click on that, fill the details and be the part of the batch. And in the Phoenix Pro batch, bache, physiology is going on, and trust me, classes are amazing, right? Each and every difficult topic is studied in such a, we are studying the topics in such a way that you cannot leave even a single PYQ from that, right? Even if you will see the PYQs after attending the class, you are going to find them very, very, very easy. Okay, so today, from today, this offer is starting and it will stay till 10th of August. Okay, so enroll in the batch now. So in the, for Phoenix Pro batch 4999, this is what you have to pay. So in the description box there is a link, click on that link. Click on that link, students, and be the part of this batch. And for getting this PDF, in the description box there is an official Telegram link. Click on that, join it, we'll share the PDF there. Okay, we will share the PDF there. So be consistent, right? The number of students are very less, they are not even consistent. So guys, if you will stay consistent, then only I will continue the classes. I'm telling you this thing. Okay.